Hello, I'm Sofie. I'll tell you something about the shape of brain structure, the genetic and phylogenetic axis of macroscale organization of cortical thickness. Cortical thickness is measured in the T1 image, MP range or MP2 range, where you measure the thickness from the gray white matter surface to the pio surface. And what you do is you have this vertex setup, this tessellation of the surfaces, and you measure the distance between corresponding vertex points in both surfaces. So with that, you have cortical thickness of the whole surface. And what you can do with cortical thickness is that you can measure structural covariance, which is a population-based measure of the covariance of seed thickness and target thickness. So for example, you take the thickness across a population of a seed region here in the anterior insula and a target region here in the sensory motor cortex, and you see if both have a correlation, which in this case they do. So using this method, you can do this for the whole cortex, where you can get a cortex-wide covariance matrix. And what you see is that regions that are relatively close to each other usually show a high correlation, and also regions with contralateral homologs show a high correlation. That's why these lines are here. Structural covariance can be largely explained by genetic factors. So if regions have a high structural covariance, it's likely the case because they have shared genetic factors. So already in 2008, using a twin sample, it has been shown that these also form certain communities which are hierarchically organized. Second, structural covariance relates to maturational coupling. So regions that show structural covariance also mature in similar ways during development. A different line of research, and we are in the gradient workshop, is gradients, which studies the large-scale organization of brain structure and function. And in a very nice paper, Daniel could show that there is this large-scale organization gradient in the human brain at rest, which goes from unimodal regions to more heteromodal association cortices here in the default mode network. And this organizational patterns corresponds also with theories of, for example, Marcel Mesulam, stating that there's this organization pattern going from more unimodal regions to higher order regions that relate to more internal processes. And indeed, like looking at the neurosynth co-activation of meta-analytic atlases, he could indeed show that in the lower percentiles along the gradient, they mostly correspond to motor processes, pain, action, eye movement, visual perception, whereas the higher ends of the gradient seem to relate more to abstract thoughts, such as social cognition, emotion, verbal semantics, and autobiographical memory. So it seems that the organization of the cortex tells us also something about the organization of cognition. So to sum up, macroscale patterns in cortical organization relate to human cognition, and cortical thickness covariance reflects shared maturational and genetic processes and is organized into multiple communities. But why? The reason for their particular spatial relationship remains unclear. So in our current study, we sought to directly examine the genetic origin of the spatial organization of macroscale cortical features. Let's go. So we used a nonlinear dimension reduction approach based on the brain space toolbox of Reine Vosterwaan, which is a great toolbox to measure gradients, and it's easy for MATLAB and Python. I used it in MATLAB, but Python is definitely also great. So what I did is I had a covariance matrix. I performed a normalized angle procedure to normalize my data, and then I performed diffusion map embedding to get the gradient ordered embedding of each region based on this structural covariance pattern, and later also based on this genetic correlation pattern. So what I found is two gradients. One goes from posterior regions, which are largely also unimodal regions, to frontal regions, and the other one from inferior regions to superior regions. Okay, nice, but nice colors, but what does it mean? So first, I also looked at how it corresponded to this organization pattern that Daniel also showed from unimodal to default mode network. And indeed, this first gradient seemed to have some correspondence to this functional organization, whereas the second one did not really do so. So yeah, nice. I've shown in this is in the ACP sample, this patterning, but then I wondered, is it actually robust? So for this, I looked at the ENKI sample and I did the same procedure. I took the covariance matrix and I evaluated what are the two major organizational axes. And again, what I found was this posterior to anterior patterning on the one hand, and inferior to superior patterning on the other. Then I also tested whether it mattered if I use FreeSurfer 6.0 or CVIT, but always there were largely these two patterning going on from posterior to frontal regions and from inferior to superior regions, or from, no, that was how it was. It was actually not, it was always the same. And also using different parcellation schemes, I also found like the Seacon, Glasser, Schaefer all largely gave this similar effect going from posterior to frontal regions on the one hand and inferior to superior regions on the other. Like, it's important to know there are small differences between these patterns, but overall they're kind of comparable. So next, I used advantage of the twin-based setup of the ATP young adult sample, and this sample includes monozygotic twins, dizygotic twins, and also unrelated individuals, which made it possible to further deconstruct this covariance matrix into genetic correlation and environmental correlation by looking at the genetic similarity of each individual and how this relates to the correlation. And indeed, as also other people have shown, we found a high relationship between structural covariance and genetic correlation, and not so much to environmental correlation, which is also something that has been mostly attributed to noise, but part of it is also likely other factors that are all put in this environmental correlation aspect of the correlation. So moving forward, I also looked at how it aligned with functional communities, and what you see is there's a high genetic correlation, sort of high, between within a functional community, but not so strong across functional communities, and a very weak but kind of similar pattern was observed in environmental correlation. So it seems that genetic correlation is also organized, and then I performed a gradient analysis to see what is the spatial organization of genetic correlation, and I found again these two patterns. So on the one hand, this posterior to frontal patterning, and the other one inferior to superior patterning. And indeed, this first gradient again corresponded to functional organization as described by Daniel, but the second gradient did not so much, or did not at all actually. So first I could show, okay, these spatial patterns in structural covariance are likely due to genetic factors because there's such a high correspondence between the structural covariance gradients and the genetic correlation gradients. However, what about phylogeny? Is there any, is it evolutionary conserved? So for this, I looked at macaque monkeys, and I took advantage of the PRIME dataset, which is an open dataset with non-human primate data, and with cooperation also with Think2 from New York, we created a covariance matrix of thickness in macaques in a similar way as in humans, and we performed a gradient analysis. And what I found was two main gradients, one going from temporal regions to sensory motor posterior cingulate regions, and the other one going from sensory motor regions to frontal regions. Well, nice. I colored them a bit like suspiciously similar to the human gradients, but are they actually comparable to the human gradients? So for this, I used a really nice approach which Ting also developed, and I transformed the human gradient to the macaque space. And using these transformed gradients, I was able to directly compare the human and the macaque gradient. And what I found is that the human principal gradient corresponded roughly to the macaque secondary gradient. So you see here there's a positive correlation between the macaque G2 and human G1, whereas between macaque G2 and human G2 there is a negative relationship, but this was not statistically significant. On the other hand, the human secondary gradient related to the macaque principal gradient. So cool, the gradient seems phylogenetically conserved, which is interesting, I guess. So using the gradient methods, we kind of were able to move a little bit forward and understand like what is the genetic basis of covariance and why is it spatially organized in the way it is. So like this posterior to anterior patterning, it relates to a functional hierarchy, and this inferior to superior patterning, like, well, I couldn't answer it. I found it in humans and macaques. It was also there in genetic correlation, but what it was still like unclear. So for this, then we looked at the theory of dual origin, which is a seminal theory stating that cortical development stems from two origins. One is in the archi cortex, the hippocampus, roughly, and it follows these arrows. And the other one is in the paleocortex, which is also the piriform cortex, and it follows these arrows roughly. So the dual origin, it relates to cortical differentiation. So it goes from low, medium to high cortical differentiation from both origins, so to say. So probing this model, I used the geodesic distance from both the archicortex and the paleocortex distance, and I wanted to see if this theoretical model would also reflect the large-scale organization I found in structural covariance. And indeed, you see when you order covariance in bins, you see that there's a high covariance at similar distance from the archicortex and also the paleocortex. Then I also looked at my gradients, and what I found is that the gradient 2 had a negative relationship to archicortex distance, but a positive relationship to paleocortex distance, possibly showing that this gradient might reflect this dual patterning, whereas the first gradient, I did not so much relate to it at all. So it's kind of a nice dissociation as well. And we could also find similar patterns using a similar approach in macaque monkeys, where we found this gradient 1, which corresponds to gradient 2 in humans, showed this inverse relationship to the dual origin distance, whereas the second gradient did not so much. So the take home is that this covariance is organized along evolutionary and functional axes. The posterior-anterior patterning relates to its functional hierarchy, whereas this inferior-superior patterning relates to the dual origin. And another take home is that I have a big thanks to open neuroimaging data, because I use the PRIME dataset, I use the ATP dataset, and ENKI. All these datasets are not scanned by me, but I was just able to take advantage of all this great open neuroimaging. So thank you so much. Yeah, an important point, like, what about distance? Because as some of you might know, distance is like an important organization principle in structural covariance. And regions that are super close to each other, they show often a high covariance, whereas regions that are distant to each other don't show such a high covariance. And indeed, if you take the gradient of geodesic distance, you see almost this highly similar pattern. So like maybe what I just showed was just pure like geometry and distance. And the same is also true for macaques, actually. So in humans and macaques, we see this, and like I was like, oh no, maybe it's not so meaningful after all. But then we regressed out distance in our covariance matrix, and what we found is these two patterns. And it's important to know that this first gradient, it corresponded both to G1 and G2, but what is important, we still find this dissociation, that gradient 2 shows a statistically significant association with Daniel's organization, whereas only the first gradient, so the gradient got reversed when correcting from distance, showed that relationship to the dual origin. Which is kind of nice, is that still if you remove the distance organization, still this functionally hierarchy and this dual origin are still present in structural covariance. And the same was the case for macaques, and even a little bit clearer maybe, where you see a clear relationship to the archicortex, paleocortex distance in the first gradient, but not so much in the second. And you see also a clear correspondence between gradient 1 and gradient 2 and the non-corrected gradient 1 and gradient 2. So this is, I hope, a happy end to a very nice project, and I was super honored to work with great people. Simon Eichel, of course, for supporting me during my postdoc, Ting, Daniel, Jajat, Casey, Alexandros, Peter, Jonathan, Thomas, Boris, like, thank you all so much for your support and your creative insights and your help with this amazing project, and thank you for listening to me.